By the time you finish watching this video, you will have aged approximately 10 minutes. That sounds trivial. But here is the shocking part: scientists have now identified specific biological mechanisms that determine whether those 10 minutes age you fast or slow, and the difference between the two extremes can be as large as 20 to 30 biological years. Not calendar years. Biological years. The kind that show up in your cells, your organs, and ultimately, your lifespan. Welcome to one of the most important conversations happening in modern science right now. In the next 10 minutes, you are going to discover exactly what aging actually is at the cellular level, why some people look and feel decades younger than their chronological age, and what the latest research says we can actually do about it. And there is one finding buried in the longevity literature that completely upends what most health experts have been telling you about lifestyle and aging. We will get to that. But first, we need to start at the very beginning, inside your cells. For most of human history, aging was considered inevitable and unchangeable. It was simply what happened. You were born, you grew, you aged, you died. The end. Ancient physicians attributed aging to an imbalance of bodily humors. Medieval scholars called it God's design. Even 20th century biologists largely treated aging as a background process, not something worth studying as a disease or a solvable problem. That began to shift dramatically in the 1960s when a cell biologist named Leonard Hayflick made a discovery that would eventually reshape everything we thought we knew. Hayflick observed that human cells in a laboratory setting do not divide indefinitely. They divide roughly 50 times and then they stop. Completely. They enter a state called senescence, where they are alive but no longer functioning properly. This became known as the Hayflick limit, and it pointed researchers towards something sitting at the tips of our chromosomes called telomeres. Think of telomeres like the plastic caps on the end of a shoelace. Every time a cell divides, those caps get a little shorter. When they get too short, the cell can no longer divide safely. The cell either dies or becomes senescent, a kind of cellular zombie that lingers in tissue and causes inflammation. This was the first major mechanistic clue. Aging was not just happening to you. It was happening inside you, one cell division at a time. And more importantly, it raised a question that scientists are still chasing today. Can we slow the shortening of telomeres? Can we even reverse it? That question opened a field of research that now involves billions of dollars in funding, Nobel Prize winners, and some of the most ambitious scientific projects ever attempted. In 1984, Elizabeth Blackburn and Carol Greider discovered telomerase, an enzyme that can actually rebuild telomere length. This discovery eventually won them the Nobel Prize in Physiology or Medicine. In theory, activating telomerase could slow cellular aging. But here is where it gets complicated. Telomerase activity is also associated with cancer cell immortality. Cancer cells essentially hijack this mechanism to divide forever. So simply turning on telomerase across the board is not a straightforward solution. But telomeres are only one piece of the puzzle. And honestly, not even the biggest piece anymore. In 2013, researcher David Sinclair along with colleagues published a landmark framework identifying 9 hallmarks of aging. These include genomic instability, epigenetic alterations, loss of proteostasis, mitochondrial dysfunction, cellular senescence, and several others. Each of these represents a distinct biological pathway that degrades with age. And crucially, each one is a potential target for intervention. This was not just an academic exercise. This was a blueprint. A map of aging itself. Of these 9 hallmarks, epigenetic alterations have emerged as perhaps the most exciting frontier. Here is why. Your DNA does not change much over your lifetime. But the way your genes are expressed, which ones are turned on, which ones are turned off, changes enormously. This is controlled by chemical tags on your DNA called the epigenome. As you age, this tagging system becomes increasingly disordered. Genes that should be silent start expressing. Genes that should be active go quiet. David Sinclair's information theory of aging proposes that this epigenetic noise is the master driver of aging itself. And if that is true, it means aging is fundamentally a loss of biological information, not just wear and tear. So what does this mean practically? It means that interventions which restore epigenetic order could theoretically reset biological age. And this is no longer just theory. In 2020, Sinclair's lab published research in which they successfully restored vision in old mice by resetting the epigenetic state of retinal cells using a combination of just 3 genes. The mice's eyes became younger. Not metaphorically. Measurably, biologically younger. The implications are staggering. But here is what nobody talks about. You do not need to wait for gene therapy to start influencing your epigenome. Because your daily behaviors are doing it right now, for better or worse. Every meal you eat, every night of sleep you get or miss, every bout of exercise, every chronic stress response, all of it is sending signals that alter your epigenetic state. The question is whether those signals are accelerating your biological clock or slowing it down. The research here is remarkably specific. Caloric restriction has been shown in dozens of studies across multiple species to extend both lifespan and healthspan. In humans, a 2022 clinical trial called Calorie demonstrated that just a 12% reduction in caloric intake over 2 years produced measurable slowing of biological aging, as assessed by epigenetic clocks. These are sophisticated tools developed by researcher Steve Horvath that can measure your biological age through patterns of DNA methylation. They are arguably the most accurate aging biomarkers we currently have. Exercise, particularly high-intensity interval training and resistance training, has been shown to directly influence mitochondrial biogenesis, essentially the creation of new, healthy mitochondria in your cells. Mitochondrial dysfunction is one of those 9 hallmarks. Which means regular vigorous exercise is not just good for your heart. It is addressing aging at a mechanistic level. A major 2017 study found that high-intensity exercisers showed telomere lengths 9 years younger than sedentary individuals of the same chronological age. 9 years. From exercise alone. Sleep is where most people are unknowingly accelerating their aging. During deep sleep, your brain activates the glymphatic system, essentially a biological cleaning crew that flushes out toxic proteins including amyloid beta, the same protein associated with Alzheimer's disease. Chronic sleep deprivation does not just make you tired. It accumulates cellular damage faster than almost any other lifestyle factor. Research suggests that consistent short sleep is associated with an epigenetic age roughly 2 to 4 years older than those sleeping 7 to 9 hours nightly. And then there is the compound that has generated perhaps more longevity research buzz than anything else in the last decade. Rapamycin. Originally an immunosuppressant used in organ transplants, rapamycin inhibits a cellular pathway called mTOR, which governs cell growth and metabolism. When mTOR is inhibited intermittently, organisms from yeast to mice to primates show dramatically extended lifespans. Human clinical trials are underway. Many longevity researchers and physicians are quietly taking it themselves. The data is not complete yet. But the mechanism is some of the strongest we have ever seen. The real-world implication of all this science is genuinely empowering. Your biological age and your chronological age are not the same number. They can diverge significantly based on what you do. And the gap between them is modifiable. People who consistently sleep well, exercise vigorously, manage stress, eat minimally processed food, maintain strong social connections, and avoid smoking have been shown in population studies to live up to 14 years longer in good health than those who do not. 14 years of additional healthy life. Not years in a hospital bed. Functional, vibrant years. The science of aging has moved from philosophy to biology to actionable intervention in the span of a single human lifetime. We now understand that aging has causes. Causes have mechanisms. And mechanisms can be targeted. We are not yet at the point of dramatically extending human lifespan through medicine, though many serious scientists believe that threshold will be crossed within decades. Before you go, if you learned something valuable today, hit that like button right now. Subscribe to Health Core Daily for a brand new video every single day. We will see you in the next one.